start our lecture of medical biotechnology, and today we will be studying about the gene knockdown. We heard about the gene knockout, and this lecture is about the gene knockdown, what that term exactly means, and how this technique was evolved, and how we can use and manipulate that technique to execute the cutting edge scientific research out there, and how it has participated and it is participating in ongoing life research in life sciences. I'm sharing my screen with you from now, and you should be able to see my slides now for the lecture. Okay, if you can see, just raise your hands so I will be able to see the people whom I can see. Okay, thank you very much. Okay, here we go. I will be going to the full screen to talk about what is RNA interference and what is gene knockdown. I really miss whiteboard for this particular lecture. I used to write a lot for these things about RNA and genes. So I will design a way, in the next lecture, how to write, and you should be able to see whatever I am writing somewhere. There are options of the whiteboards here on the screen, but they're not very well controlled form of writing and drawing things. I'm not very good in drawing that. Anyway, so let's start with our today's lecture formally, that is about the RNA interference. If you people, you definitely know this diagram. This is supposed to be the central diagram of the last century. This is like the central pillar of life sciences. It actually explained everything, how life works. Life functions with the help of wonderful molecules called proteins. These are functional components. And these proteins are created out of the messenger RNA. Messenger RNA brings information, and they are created out of the DNA using the information present on the DNA. So the most fundamental question of life sciences, that how DNA can control the function of life, can be answered with this diagram. And that is why it is called as the central dogma. And it's too simple to ask that DNA is like the center of all life activities and it can control the life present in the cell, but how? The answer lives here. DNA can express to produce messenger RNA, and then this messenger RNA is translated to produce protein. And remember, having a piece of DNA means you will be able to generate certain pieces of RNA, and those RNA will be translated to produce certain types of proteins. Having a bacterial DNA will make you a bacteria, and this is not true. Am I right? We are carrying a lot of bacteria, but we are not bacteria. We are carrying a lot of viruses as well nowadays, but still we are not viruses. We are carrying human DNA, but somehow some of us is not human as well. So that is something we always wondered, that how DNA can be categorized as something which can be referred as a material which is controlling the life. It's not about having the DNA. That's what I wanted to tell you. Otherwise, many of us might be viruses today, because we are carrying a lot of viruses in our genome. It's not about having DNA. Having DNA means having certain RNA. Having certain RNA means having certain proteins. So it's more about how to control the DNA to produce the messenger RNA of its own choice. to have proteins of its own choice. It's not about having DNA, it's about how we can control the function of DNA. Otherwise, we have, for example, 1,000 genes, definitely more than 1,000. But think about if we are having 1,000 genes within our genome, those 1,000 genes are kept on expressing and kept on producing RNA. They will be kept on translated. They will kept translated into proteins. So we will be having 1,000 proteins always in our each and every of our cell. In that situation, we will be having a life form with zero variation. We will be having a life form which is exactly identical with everything else. 1,000 genes, 1,000 proteins. But that life is not, our life is not like that. It is like striking each and every keyboard, each and every key of the keyboard of a piano. If you strike each and every key of the piano keyboard, you will end it up in something which is very constant for everybody. Everybody hits all the keys at the same time and keep hitting it will produce like the same tone every time. But the DNA somehow evolved to find a way that how to express a certain messenger RNA and how not to express certain other messenger RNA. This is called regulation of gene expression. And this is what life is all about. So having a certain piece of DNA means a certain pieces of RNA is not true. The true version of life means having a certain piece of DNA and then the mechanism that how to control that DNA to produce a messenger RNA of your own choice. That is why one cell type is different from another cell type, because one cell type chooses to express a different combination of messenger RNA from the same DNA, and the other cell will choose to express another type of combination of messenger RNA using the same DNA. Each and every cell we are carrying have the same DNA. But the control and ability to express the same DNA in a different way is actually the seed of life out there. Is actually what life is all about. Otherwise, putting bacterial DNA in something is not enough to be like a bacteria. Putting a human genome in somebody's cell is not enough to be human. It's about how we able to control the expression of our genes. The gene knockdown system and the understanding of the gene knockdown system will help you to understand one of the fascinating ways the nature has evolved over the period of millions of years to control the expression of genes in one way or another. So having DNA means RNA, having RNA means protein. But now having DNA means the RNA of cellular choice. That is called regulation. Having the same DNA with different types of messenger RNA, different compositions of messenger RNA, different concentration of messenger RNA, and then produces different types of proteins. Certain other terms which I have already explained earlier in the class, I would like to repeat it once again to refresh your memory. The genome contains large number of genes, and the genes are that part of the genome which can be expressed to produce messenger RNA. That does not mean the other portions of the DNA are not transcriptionally active. The other portions are also expressing RNA, but those RNA are not messenger RNA. They might be transfer RNA, they might be antisense RNA, they might be long non-coding RNA. But whatever those are, those will not going to be used to produce proteins. Only one type of RNA is able to produce protein, and those are called as messenger RNA. So the DNA is carrying lots of genes. The protein coding portions, some of the portions produce, the protein coding portions can produce the messenger RNA. DNA can control, first, the DNA can control whether it is required to produce a particular messenger RNA or not. DNA can choose whether I need to express that gene or not. That is the first level of the regulation. This is called transcriptional regulation, or the regulation at the transcriptional level. If you remember, if a cell receives a particular transcription factor, if a cell is carrying a particular transcription factor, it will start expressing certain target genes. If another cell is not having the same transcription factor, the other cell will not going to express the same target genes. And that is why we will be able to have two different cells from the same DNA. So first thing, the cell can choose to express a different combination of genes based on the presence and absence of the transcription factors and the conditions. So that means the cell can choose whether it is required to express a certain gene or not. It is called regulation at the transcription level. Okay, I wish you should remember whatever I'm telling you. Okay, this is supposed to be very important. First level of regulation. Once the messenger RNA is created, my hands are, so much anxious at this time to write. The second, I'm trying to create images in your mind using my words. It's not easy, but I'm just trying. Okay, just please help me. If you're not getting the things perfectly, clarify it with the help of your questions. Okay. Once the messenger RNA is there, the nature still holds the capacity to control and tune the levels of messenger RNA within the cell. Whether that messenger RNA is exactly required or not, is this messenger RNA is fine or not, is this messenger RNA should be kept or not, if this messenger RNA is not required. So nature still can manipulate the messenger RNA based on the importance, based on the requirement. This is called post-transcriptional modifications, or the control of gene expression at a post-transcriptional level after the transcription. Still control it. If a messenger RNA is really required, it is changed structurally so that it can live. If the messenger RNA is not required, it is produced because of some XYZ reasons, and cell is not interested in having that messenger RNA. It is manipulated, it is changed in such a way that it is destroyed. Or if a cell do not require the expression of a certain messenger RNA with that much concentration, the cell can tune it down with the help of certain modifications called as post-transcriptional modification. And this type of regulation exists at that level, and it is called regulation at the post-transcriptional level, after the transcription. Once we have the messenger RNA ready to be transported into the cytoplasm, now it's gone. The message is there out of the cytoplasm. The cytoplasm contains ribosomes. Ribosomes will capture those messenger RNA and will start producing the protein. But no, the control is still there. Even at that time, cell can control whether to translate that messenger RNA is absolutely necessary or not, whether it is required to be translated or not, because it's a very huge activity consuming lots of resources. अगर हमारे सारे genes express होके translate होना शुरू हो जाए, we will be dead in minutes. We don't have enough resources to translate everything. We just need to translate whatever is absolutely required in the context of that cell. For example, if the cell is in trouble, the P53 is required to be expressed. The cell is not in trouble, it will going to be a waste of energy to produce those proteins. The cell, what will happen after that? The regulation occurs at the translation level, whether the translation is required or not. And even the protein is created after the translation, the cell still holds another level of regulation, which is called as post-translational modification, or the regulation of gene expression at the translation level or after the translation level. If the cell is not required, if the protein is not required at that time, and it is created, but it is not required. So what the cell is doing, the cell will put a certain modifications on the protein structure, so that protein will be inactivated but present. So when the function is required, those modifications will be removed. Those modifications will be removed, and then the protein can be used for its function. Think about the P53. It's a tumor suppressor required to be used when the cell is under stress for many reasons. It's very critical and important protein, but it is not required always. So it is, the messenger RNA is created, the proteins are created, but once the protein is created, it is kept inactivated with the help of modification using MDM2 and other things. So when the trouble is there, only the modification is reversed and the function of the protein is restored. That quick. It is not good at that time if the trouble is there and the cell was pushed to create that protein. The cell will be very late if it start the activity from the DNA level. Cell कहेगा DNA को RNA बनाओ, फिर RNA से कहेगा protein बनाओ. It will take a lot of time. The protein is there, just need modification to get the function back. So all these modification actually controls that how a certain cell will be working, and this is called the regulation of gene. And this is life. The actual definition of life is the control on the DNA. That how we can manipulate the DNA, how we can control the DNA with our own choice. If you do not want to have the function of a certain gene, because genes are there, if you do not want to use a one particular key on the piano keyboard, what is the way? What is the way not to use that key? I am answering on your behalf, okay? I'm just assuming that one of the students will answer this. The other student will answer this. I'm missing your responses. So what are the ways that we just don't want to use a one particular key in our piano keyboard? First way, the key, you should destroy that key. Now hoga baans, na bajegi baansi. If the key is not there, you will not be using. But imagine this is not the very best way of not having a function, since kal ko, parson ko, in another situation, you might want to play that key. In another situation, you might be needing the function of that particular key. So destroying that particular key is not the very best of them. But destroying of that key means, which we studied in our last lecture, that means gene knock out. Killing the gene, destroying the gene of not having the function. The gene is not there, the function is not there. But this is an irreversible thing. Nature cannot use that way. Because if nature do not want the function of a particular gene, and nature choose to destroy that gene, and what if the function of that gene is required after some time. It will not be, the nature will not be able to have the function again. Whatever the genes we are carrying, remember, whatever the genes we are carrying, these genes are the product of millions of years of evolution. They are required in one way or another. We are like the best product, Ahsani tak meme. We have been created like this. So they are required one way or another. So nature designed a very wonderful mechanism how to control the expression, how to control the function. And that technique is called as the knockdown, which we will be studying. So if you want to control the function here, you should delete the protein. So nature created a certain small strands of RNA, small strands of nucleic acids or RNA, which can complement itself with a certain messenger RNA. This complementation leads to the destruction of a certain messenger RNA. The messenger RNA is gone, the protein will not be created, the function will be silenced. If you want to restore the function, you can choose not to express those small interfering nucleic acids. So with the help of interfering nucleic acids, nature controls whether a certain messenger RNA is required or not, and if it is required, in how much concentration. Now let's see how this wonderful phenomena came into existence in the history. Like every other scientific discoveries, that was also an accident. When scientists started studying about the genes, DNA, RNA, and protein, when they started learning about these things, one of the scientists at that time, he wanted to confirm his hypothesis. You should see those papers which I have mentioned. You are not supposed to memorize those papers, but I want you to at least write 3 or 4 lines out of each and every of the paper which I will be mentioning in this lecture. That will help you to grab something out of those papers. I have collected those papers for you which are very relevant for this topic. So somebody was working on a gene which imparts this brilliant red color to a plant. It's a botany stuff. Dr. Rehan might be wonderful in that. Petunias, which actually imparts this bright red color to the plant. He is working on a gene. He knows that gene is responsible for this color. So the scientists wanted to enhance the brightness of that gene. So they have created an RNA, a gene, actually a gene. They have created a nucleic acid in some way, using some way, and then they insert that nucleic acid within the cells of that plants, assuming what? Giving more gene means will be more expression, will be rich and more bright colors. But unfortunately, when they inserted that gene from outside to the cells, it decreases the color. When they inserted more, it actually removed the red color out of the flower. So the results are entirely opposite what these people were expecting at that time. It was such a mysterious outcome. They were expecting the color to be more brighter, and they were observing entirely opposite realities. The colors goes down when they add more and more gene. Now you can guess what was happening at that time. Guess what was happening using the earlier slide. Whatever the genes they were sending to the cells, those genes are complementing with the messenger RNA. They were assuming they were giving the genes, but unintentionally they were giving the strands which are complementary with the actual messenger RNA of that gene. whatever they are sending, it is actually destroying the messenger RNA and decreasing the outcome and the function. So these are the follow-up papers with the same thing. Some people, these people, ये मुझे लगा जैसे Dorothy है. I wish you people know the Dorothy. If you don't, then you are not very good in animated movies. So these people try to reduce the expression of a virus with the help of certain gene. They want to study the function of the virus. So they give the gene of the virus to the tobacco, and what they found, the co-suppression. They were assuming that I am giving the gene of the virus to the tobacco, it should increase the viral genes, it should increase the viral expression, but they found the co-suppression. So initially it was termed as the co-suppression, giving certain gene which reduce the expression. They have no idea about that thing at that time. And then these people actually triggered and the activity by giving the small RNA, the small RNA. Later they came to know the small strands of double-stranded RNA, when given to the cells, they can silence the function of one particular gene. It was such a beautiful and wonderful discovery of that time that it generated a Nobel Prize out of their work. They have designed a system that by designing a double-stranded DNA of our own choice, we can selectively destroy one particular messenger RNA. It was such a wonderful, such a general and great technique of that time. It's still working. I mean, imagine the possibilities. If you do not want the function of one particular gene, just create the double-stranded RNA or double-stranded DNA. That double-stranded DNA will enters into the cell, will be processed. I will explain you how. Will be processed and will eventually destroy the function of a particular gene of your own choice. There was no DNA damage. Remember, in this thing, the DNA is intact. Nobody is destroying the key. Nobody is destroying the original DNA. And what we are doing, by giving the double-stranded DNA of our own choice, we are killing the messenger RNA of our own choice. Okay, so that is how we can manipulate and control the function of life by sending something right within the cells. And I'm telling you, that might be the first time when we able to control life in a real sense at a molecular level. I mean, it's not like controlling science like a sci-fi movies where you can actually ask somebody to move the hands, when you ask somebody and control somebody to start running or start sitting or start doing things on your choice. At a molecular level, that is how you can control the life, by sending something and destroying the function of messenger RNA. This is called knockdown, by knocking down the messenger RNA and then deleting the function. You can reverse it and restore it whenever you want. You just need not to give the double-stranded DNA. Once you're not giving the double-stranded DNA, what will happen? The function will not be down-regulated and the function will be restored. That is why these two people were given for their work with the RNA interference. So there's the gene, produce messenger RNA. If you destroy the messenger RNA, the function will not be there. It's called RNA interference. It gradually evolved over the period of time. I want you people to please try to write like 3 or 4 lines with your own wisdom after scanning or reading these papers. That will help you to understand how that story develops over the period of time. It does not end it like this, that they came to know that I will be sending the double-stranded DNA. It did develop like the first experiment which I shared you. They send the DNA, it destroys the function. First hint, something we can send can go and interfere with the function. And then they started investigating the reasons and ended up like the DNA can and external genes can interfere. And they came to know that a duplex of 21 nucleotides might be enough to downregulate the function of the genes. And this study has published and showed that when people give the duplex of 21 nucleotides of their own choice, they're able to downregulate the function of the genes. Okay. These blue spots, they are actually the cell cultures, the cells, okay? The blue ones are the nucleus. It is stained with DAPI or Hoist. They are the dyes which can color the nucleus. Papers में अगर इस तरह की pictures हो, तो ये reviewer को convince करने के लिए होती हैं कि I am talking about the cells. Whatever you are looking at, these are cells and these are the nucleus. And green is one of the green color antibody against one particular protein, and red is another antibody against one particular. This might be the control and this might be the knockdown of a particular gene. The expression is reduced when they added the duplex of 21 nucleotides. They discovered that mechanism and how it actually works at a higher level in the mammalian cells as well. So the process is called as RNAi, means RNA interference, using the small duplexes of nucleotides. And they work post-transcriptionally. Am I right? Now this term should be understood. They work after the transcription. And it executed in two key steps, which I will be talking in the next session. Okay, so you people are supposed to have a break. I will be sharing a small video with you. But okay, then I'm not sharing. I will be talking and you can see the video later on as well. Okay, as long as you can do that thing later on as well. So in the next session, I will be talking further about that mechanism. I hope this session helps you to develop that what we will about to see in the second phase of our class. Okay, and remember, the online classes and online system is not very ideal ways of learning right now, because since we were not trained, we were not smart enough like you to control and to internet and to deal with all these smart techniques out there. But somehow we are finding a way and we are trying our best to to deliver you the appropriate knowledge. I can tell you I'm missing my whiteboard and writing things a lot. I will be finding a way. Okay, मैं कुछ चक्कर चलाता हूँ. Don't worry, I want to speak on these things. Let me record that video for you, and let's restart the second session for about understanding about the regulation of genes, and in specific about the gene knockdown. So let me share my screen once again with you, so that you can see that I have made a smart move for you people. Can you see my page? Can you see my hands? Okay, can you see my pen? Okay. I wish that the internet speed helps me to write in real time for you what I was talking about. Okay. So if everyone is ready with me, so I want you to understand one thing, that a gene. Okay, I don't like this page, frankly. This has the lines. I don't want this page anymore. I don't have the other page as well. Okay, let me use something more. So this is supposed to be the DNA strand. This is supposed to be the DNA strand. Okay, this is the DNA. It's constant for every cell. It's almost constant between us as well. And all of the trillion of cells we are carrying, the DNA is a constant thing. That DNA contains a very small fragments, very small portions, which we call as the genes. These are the parts of the DNA which are capable of producing the messenger RNA. The symbol of messenger RNA usually a certain number of A's at the end, which is called as poly A tail. So any portion of the DNA is a gene if it is able to express a messenger RNA. It may be messenger RNA name 1, this is the messenger RNA name 2, gene 1, gene 2, this is the gene 3, and this is producing the messenger RNA number 3. So one part of the DNA, one part of the DNA is called as genes when it is able to produce the messenger RNA. But remember, this gene constitute less than or almost 2% of our total genome. So what that means, there are a lot of places which are not expressing anything, which are not producing protein coding messenger RNA. So initially people thought those things, those places which are not producing anything, they are like the useless things. If the video is stopped, just to one of you can let me know, okay? Just turning on your microphone. It seems it has stopped rendering. Okay. It's telling me that my internet connection is not well. So the portion of the DNA which is able to produce a messenger RNA is called as the gene. That does not mean that the portions of the DNA, which are not producing any messenger RNA are not important. Later on, people discovered that actually these are the places from where the other genes are regulated. These are the places from where nature controls that how other genes will be expressed. Out of the many things, there exist certain portions which are considered as genes, but they are producing an RNA which is not similar to the messenger RNA. When I said this is not similar to the messenger RNA, that means it does not contain the bona fide signature marks of a messenger RNA. It will not be captured by the ribosomes. It will not be used to produce proteins. Although RNA pol 2 is transcribing that gene to produce messenger RNA, RNA pol 2 is driving that gene as well to produce an RNA. But that RNA is not going to produce any protein. Both of them are produced from the DNA using the RNA pol 2. Now, one of the RNA is called messenger RNA, since it can be captured by the ribosomes and translated into a protein. And the other type of RNA, which do not contain any marks, any signatures similar to messenger RNA, and will not be captured by the translation machinery and will not be used to produce protein. Those genes are called as known coding genes. They are also genes, but they are called as the known coding genes. And that part of the genome is called as the known coding portion of the genome. They cannot produce proteins, but they can control the function of other messenger RNA. They can control the function of other messenger RNA. So this is the significance of the known coding part of the gene. The things which I talked earlier, the double stranded RNA, they are actually hacking. They are mimicking a natural way of controlling the expression of gene. They are mimicking a natural mode of controlling the gene expression. Now, if you are clear about the known coding parts, known coding portion is able to generate the RNA, but that RNA is not a messenger RNA. That RNA might be of different types. That RNA might be of different type. It might be a long non-coding RNA. It might be a small interfering RNA. It might be a micro RNA. ये term आपने सुना होगा, micro RNA. This might be a micro RNA. Imagine the micro RNA produced from this site. I am referring this site as X. X-Men देख रहे हैं आजकल तो दिमाग में वही चल रहा है. I am referring this site as the X gene. The X gene is able to transcribe to produce an RNA, but that RNA is not a messenger RNA, which means it will not going to produce any protein. But I'm telling you, it is producing a micro RNA, a small fragment of RNA. Now, luckily, if the sequence of that micro RNA matches with the sequence of messenger RNA number 2, what you will expect? The synthesis of gene X, the transcription of gene X will produce a microRNA, which will eventually, this is not going to happen in one step, okay? I'm just trying to be very simple on that and very gentle and very kind on gene regulation. The transcription of gene X generates an RNA, which is not a messenger RNA. Actually, it's a microRNA, for example. And this has a complementarity with the gene number 2. So what you can expect, as long as the transcription of X is going on, the transcription of gene number 2 will be silenced. It will going to block the function of gene number 2. Now this gene is regulated by the gene number X. This gene is controlled by another transcription factor. This gene is controlled by another transcription factor. And the balance of these two transcription factors defines how much the product of gene number 2 will be there. The beauty of regulation is that the gene knockout will give you, a choice of having a gene or not having a gene. That's all. Whether you will be having a gene like a turn on and turn off. But the regulation, as I explained you, at a messenger RNA level will give you a huge option to regulate the expression of the gene between two big ranges. Like the regulator of the fan. If the fan do not have a regulator, you can either turn on the fan with full speed, or either you will not going to have the fan at all. But if you have a regulator of a fan, you can control it between different speeds. That is how we able to have the function of a certain gene in different grades. The brilliant or beautiful level of transcription. Let me take you quickly in the next 10 minutes or 5 minutes about the mechanism, that how the double-stranded DNA, the model works, and how it actually stop and destroy the function of the gene. I will be sharing a video with you, and you can see the videos as well from the internet, which will explain you beautifully the mechanism. For me, it is more important for you to understand the logic behind having that function. So once you have the double-stranded nucleotide sequence, when it enters into the cell, our cells has a built-in mechanism to chop off and destroy a free double-stranded nucleotides. So they should not exist within the cells. They might be viruses. They might came from another bacteria. So our cells use a machinery called dicer. That machinery chopped off and destroy the double-stranded nucleotides into simpler, smaller fragments. They are called as small interfering RNA. This small interfering RNA later on will be utilized by a machinery called RNA silencing complex, RNA interfering silencing complex, and one fragment of that interfering double-stranded nucleotides, only one of the fragments, see the red one and the blue one, the double-stranded nucleic acid, only one of the fragment like the blue one is used and it is loaded on the RNAi silencing complex. Now if that blue part of the nucleic acid has a complementarity with any messenger RNA, if the messenger RNA finds its complementarity with the blue sequence, the translation of the messenger RNA will be halted or the messenger RNA will be destroyed. So that is how a double-stranded DNA of your choice will produce a small interfering RNA of your choice, and it will target a messenger RNA of your own choice. That is how it can destroy and silence the function of a certain gene. And scientists have shown that 21 to 23 nucleotide fragments of double-stranded RNA is good enough. It will be destroyed, not destroyed is not a good word, chopped off with the help of the proteins called dicer into small fragments. They are called as small interfering RNA, and then these short interfering RNA can silence the function of the gene. Now think, if you are able to create those small interfering RNA by yourself, you can control the function of any gene. And this is what the gene knockdown technology is. When I have done my job as a PhD student, I have created the small interfering RNA to silence the function of one particular gene of my own choice. So the long double-stranded RNA chopped off into simpler fragments, those single fragments of about 21 to 23 nucleotides having 19 nucleotide duplex and a few base pair overhangs. That is how nature created them. So we need to mimic in the same way. And those duplexes, like this is the duplex of small interfering RNA. One of the fragment is loaded on the complex. I told you the name, the RNAi silencing complex. Or sometime, but not sometime, but mostly, one of the fragment is able to bind itself with the target messenger RNA. If that complementarity occurs, the messenger RNA will not be able to survive, it will be chopped off. The complex and smartness in this procedure lives here. अगर ये समझ आता है तो काम पूरा हो जाएगा. There are two strands on this double-stranded DNA. Actually, it's a double-stranded RNA. The one is referred as blue, the other is red. The blue one has the same sequence of the target messenger RNA, and red one automatically will have the complementary sequence of the target messenger RNA. You need to design the small interfering RNA in such a way that the red part, the red side, the red strand should be loaded on the RNAi silencing complex. So eventually the complementation occurs. Think about the miss or a wrong designing of small interfering RNA. If you design the small interfering RNA in such a way that red one is gone and green one is loaded on the RNAi complex, it will not going to create any complementarity because it's the same sequence, and that will not result in the breakdown of the messenger RNA. Okay, I will show you the things in a little bit more of the detail. Okay, here this is another example, just forget it. And this is forget about these two slides for the time being. Okay, how we can install the small interfering RNA within the system, how we can send the small interfering RNA within the system. The one and the easiest way is that you can create the double stranded nucleotides and you can just try to insert those double stranded nucleotides within the cells with the help of certain molecules, with the help of certain chemicals. You pack those double stranded nucleotides and thrown out within the cells. Once they are within the cell, they will be chopped out into simpler components called small interfering RNA. And in the second stage, one of the fragment is loaded on the RNA sequencing complex and will find a complement messenger RNA. Once the complementation is done, the messenger RNA will be destroyed. Okay, if you have any question at this stage, I would like to answer, since the time is not enough to cover everything of this lecture. I would like to talk more on the remaining part of that lecture in our next session. So I am holding this lecture at this stage to take your questions. So whatever we have studied at this time, in the next session, I will be teaching you the detailed mechanism that how these two steps are executed, how the double stranded DNA is chopped off into siRNA and how siRNA will be loaded and then execute to destroy a one specific messenger RNA. Also, I believe the video will serve the purpose. But I will be talking on these things further. Okay, so I am stopping that lecture at that stage and will be taking your questions so that we can have an interactive session. उमर तुझे शर्म आती है बोलते हुए, टेक्स्ट करता है. So speak. I want to listen to your voice. I miss you. क्या सवाल है आपका?
Sir, I wanted to ask a question.
जी शाबाश. उमर है ना? Okay.
नहीं सर, मुस्तफा.
उमर ने क्वेश्चन पूछा है, पहले मुझे उमर की आवाज सुनने दो. आई एम स्टॉपिंग माय स्क्रीन फॉर यू जस्ट टू रीड द चैट वेल. उमर आप अपना माइक्रोफोन ऑन करें. शाबाश.
सर ऑन हो गया. अच्छा सर, my question is, what will happen if siRNA sequence is homolog to target messenger RNA?
If they have the same sequence, they will not going to bind. They will not going to destroy the messenger RNA.
Okay. And sir, is this process ATP dependent or independent process?
No, it is an energy requiring process. It's a natural defense mechanism. हम ना cheat करते हैं, हम अपने सेल के सिस्टम को चीट करते हैं. हम डबल स्टैंडर्ड डीएनए देते हैं. हमारा सेल समझता है कि ये कोई एक्सटर्नल डीएनए. They started cutting it off, and then after that it ended up like small interfering RNA. तो हमारा जो RNA interfering complex है वो बनते हैं and they start destroying the messenger RNA. So our own natural defense mechanism is fooled by the double stranded RNA which we are giving. हम इसलिए देते हैं ताकि वो हमारी मर्जी का messenger RNA destroy करते हैं. Okay? And it's an energy requiring process. जी मुस्तफा आप कुछ पूछना चाहेंगे?
Sir, I wanted to ask, are we going to study that how the siRNA from the single stranded RNA from the duplex in further slides? मतलब अभी बताएंगे कि इसको सिंगल स्ट्रैंडेड में किस तरह कन्वर्ट करते हैं ये siRNA को.
अभी से मतलब है कि हाँ जी मुस्तफा इस लेक्चर का वो हिस्सा है, part of this lecture, but since we have like run short of time, so that is why I'm not going further, because otherwise it will like create a mess. Let's hold this lecture here, and in the second part we will study the detailed mechanism and the remaining that how actually the double stranded DNA converted into single and then it is loaded on the complex and then go and find. Actually I will be giving you a video that will answer your question as well. मुझे जरूरत नहीं पड़ेगी बोलने की इंशाल्लाह, लेकिन
okay, sir.
we will love to talk अगर हमें लगा पूछना चाहिए. जी शाबाश. इससे पहले कि सेशन एंड हो, you people can keep on asking questions if you have.
Sir, I also had another question.
जी शाबाश.
Sir, I also had another question. So, what determines that which specific part of the chromosomal DNA will form a heterochromatin structure or a euchromatin structure? Because Actually, it is the euchromatin or the heterochromatin part that determines the expression of a specific DNA gene.
It is defined by the different markers present on a particular DNA. How DNA will be packed, convoluted, or create the three-dimensional architecture. It is defined by the presence and absence of certain marks. Those are called as histone marks. It is not part of that lecture, but just I'm answering you. They are called as histone marks. The presence of such marks allows the DNA to fold into a conformation which you can refer as heterochromatin in accessible form. So it is called, अगर आपने एक खतरनाक लफ्ज सुनना है मेरे से, this is called epigenetics. Okay, and do study. मजे की चीज है ये. और फरहान बोलो, I want to listen to you यार. You wrote your question. Please turn on your microphone and speak. यार मैं मिस कर रहा हूँ आप लोगों की आवाज को. शाबाश बोलो.
Yes sir. So मैं पूछा था कि RNA इंटरफेरेंस का जो है वो हम जिस तरह वायरल प्रोटीन्स जो वायरल जीन्स होती हैं उनके लिए यूज कर सकते हैं. कोरोना वायरस के लिए स्पाइक प्रोटीन है उसके जीन के अगेंस्ट साइलेंसिंग कर सकें जब बॉडी में एंटर हो जाए तो.
शाबाश फरहान, very good question, very relevant. That we are like experiencing an RNA virus right now, the pandemic. Is that possible that we can interfere that RNA with the help of this technology? The answer is yes, we can do it. But the next question is that whether it is like practically doable, like for like a drug, like a treatment. The answer is no. Originally, people thought when the interference technology came into existence that we can control everything. So nature has its own ways. हमें लगा हम सब कंट्रोल कैंसर को डील कर लेंगे. अगर कैंसर XYZ जीन की वजह से हो रहा है, we will create a mixture of interfering RNA and give to that person's tumor, and then what will happen? The gene will be silenced and person will be fine. But things are not that simple. I mean, imagine if you have created an RNA interference molecule, you need to supply that molecule where? To the right cells. Am I right? So in case of the pandemic and the coronavirus, you need to supply that RNA where? Small interfering RNA to where? To the cells of the lungs, infected cells. You cannot have a pill. You cannot get injected because the small interfering RNA is like too fragile to exist like a drug within the body. You cannot eat it through the pill. अगर आप उसे खाएंगे तो क्या होगा? तबाह हो जाएगा. So if you able to supply, अगर आप किसी सेल के अंदर इंजेक्ट कर दें small interfering RNA opposite to the pandemic virus, coronavirus, it will be destroyed. But it is practically almost like it cannot be used like in this way. Shaba, that's a very good question. आप लोगों का जो क्वेश्चन आंसर सेशन होता है ना, वो एक रॉक स्टार वाली बात है. हमारे मुल्क में साइंस की कोई इतनी कदर नहीं है, वरना जो आप लोग बातें करते हैं इस वक्त ऑनलाइन जो सवाल पूछते हैं, आई मस्ट टेल यू, आई मीन किसी जमाने में जब हम मुल्क में स्टूडेंट्स के सवालों को शाबाश देना शुरू करेंगे, जब हम इस बात की अहमियत को समझेंगे कि जब आप बात पूछते हैं तो उससे कितनी अच्छी जनरेशन ऑफ नॉलेज होती है. These sessions will be praised. Okay, you are the silent warriors जो नजर नहीं आएंगे. Okay, शाबाश. Next question. Okay. किसी का माइक टर्न ऑन नहीं. मुस्तफा का दिल कर रहा होगा, हालांकि उसका माइक टर्न ऑफ है. ओके, किसी के पास सवाल नहीं. लाइक के पास भी सवाल नहीं है. कोई लाइक देर से है?
यस सर, एक्चुअली है एक छोटा एक सवाल है, वो पिछले क्वेश्चन की एक्सटेंशन है.
हाँ जी.
अच्छा सर, वो आप अभी बता रहे थे कि देयर आर सर्टेन स्पेसिफिक मार्कर्स जो डिटरमिन करते हैं कि ये पोर्शन बनेगा हेटेरोक्रोमेटिन और यूक्रोमेटिन. लेकिन सर पूरे हर सेल के अंदर डीएनए तो सेम होते हैं, एग्जैक्टली सेम होता है, तो फिर सेल मुख्तलिफ डीएनए किस तरह एक्सप्रेस करता है?
ये एक चेन रिएक्शन का एंड है, जिसका स्टार्ट सिंपल डिफरेंसेस के साथ होता है, जिसको हमने पहले स्टडी किया है. ये एपिजेनेटिक्स है, वैसे इसका टॉपिक नहीं. अल्लाह करे कि हम इसको किसी दिन तसल्ली से बैठ के पढ़ें. लेकिन याद रखें कि जो डिफरेंस बिटवीन द सेल्स है that is defined by the difference in the pattern of expression. And that difference is initiated during the development and after that with a very small differences at the initial level. It's like a chain reaction जो बाद में start होती है. Okay? शाबाश. और इस पे बात कर सकते हैं अलग से ये इस just ज़ैनब ने अनम्यूट किया है. ज़ैनब आप कुछ पूछना चाहेंगे?
Sir, could you please discuss the role of the RNA silencing complexes? Is it responsible for segregating the two strands of the double-stranded siRNA and then complementing the desirable strand with the part of the mRNA that needs to be silenced or deleted?
Yeah, Shab. You know why I'm happy with this question? Because this question belongs to the second part of the lecture. That indicate that how much you want to know what will happen next. And that is, by the way, is you can I will be happy with this question because you want to know अच्छा आगे क्या होगा ना. अच्छा, how the double-stranded DNA got separated, RNA complex कैसे उनमें से किसी एक को पकड़ेगा. So after the lecture you can get the video and you will get the answer. Or otherwise हमारे next session में हम इसी पे बात करेंगे detailed mechanism पे कि ये होता कैसे है. Okay, याद रखें double-stranded small interfering RNA is unstable. And when you separate the two strands, the single-stranded RNA, small size, no chance of survival. So you need a protein complex which can protect it. वो हम पढ़ेंगे, वो अगले वाले लेक्चर वो प्रोटेक्ट करता है और फिर मैसेंजर आरएनए को तलाश करना, कंप्लीमेंटेरिटी को ढूंढना एंड देन डिस्ट्रॉय द मैसेंजर.
थैंक यू सर.
जी हमजा आपका भी अनम्यूट है माइक.
जी सर, मैंने पूछना था कि सर आपने पिछली स्लाइड्स में कहीं मेंशन किया था कि जीन उन्होंने प्रोटोनिया पिगमेंट की जीन प्रोटोनिया के अंदर डाली थी और उसकी वजह से उसके पिगमेंटेशन कम होनी शुरू हो गई थी.
जी जी.
तो सर अगर जीन जो है वो आइडेंटिकल है टू द पिगमेंट दैट इज ऑलरेडी प्रेजेंट तो वो उसके कॉम्प्लीमेंट्री तो नहीं होगा एंड देन इट शुडंट टेक्निकली साइलेंस द जीन.
या श्योर, गुड पॉइंट. वो भी यही समझ रहे थे, उनका भी यही मसला था ना जो आपने कहा है. वो कहते हैं हमने जीन डाला डबल स्टैंडर्ड डीएनए तो ये कम कैसे हो गया. द रीजन व्हाई दे हैव इंसर्टेड द डबल स्टैंडर्ड स्ट्रैंड, तो सिस्टम ने क्या किया, सिस्टम ने चॉप किया होगा, उसके बाद डबल स्टैंडर्ड्स को अलग अलग किया होगा, स्मॉल इंटरफेयरिंग आरएनए बनाया होगा, दोनों स्ट्रैंड्स को अलग किया. अब वो जो दो स्ट्रैंड्स को अलग किया उनमें जो बच गया दैट वाज द वन जो कॉम्प्लीमेंट्री था. दैट इज समथिंग दे हैव नो आइडिया एट दैट टाइम. You know, we have the double-stranded portion of the DNA. We don't know which of the strand is actually transcribing sometimes. So whatever left was the one small fragment which is exactly opposite to the messenger RNA. Or according to their understanding, since they don't have any interference concept at that time, they just know this is a gene. I am sending the gene. And let's see. And actually, what was happening at that time? The double-stranded gene they are sending, it was actually taken up by the cellular defense mechanism, destroyed by the cellular defense mechanism, produced small interfering RNAs, and one of the small interfering RNA got separated and loaded onto the RNA complex, and it actually complement to the messenger RNA. Happens to be complement to the messenger RNA. So वो वही सोच रहे थे जो आप और मैं और हम सब इनिशियली सोच सकते हैं कि यार एक जीन है जो किसी ने मुझे कहा है कि it is responsible for the red color. I am sending that gene to the cell. तो क्या होना चाहिए, उसको बढ़ना चाहिए, लेकिन सेल्स उस जीन को अकोमोडेट ही नहीं कर रहा. सेल्स आर एक्चुअली डिस्ट्रॉइंग दैट जीन. इट्स अ वेरी इंटरेस्टिंग एक्सपेरिमेंट. दैट वाज वेरी इंटरेस्टिंग. जी जी शाबाश. ओके, किसी का अनम्यूट नहीं हो रहा, इसलिए इट सीम्स वी आर डन विद आवर क्वेश्चंस. लास्ट सेशन वाज वेरी वंडरफुल. मैंने उसको डाला उसके ऊपर यूट्यूब पे. याद रखें कि जब तक हमारे टाइटल्स बहुत फैंसी नहीं होंगे, नोबडी विल लाइक टू यू नो सी आवर वीडियोस. Our videos are very boring. कदेश कुछ करे कुछ सोचे तो शायद हमारे लिए अपनी वीडियो को वायरल करना जो है वो आसान हो जाए. It's not possible. Remember, if I got rich, I will be sharing my money, okay, with you. Don't worry. And thank you very much, all of you, to be here. I can